Good afternoon, everyone. I welcome all viewers to the 74th Vyakhyaan in the PRL का Amrut Vyakhyans series of online lectures or Vyakhyans. This series of 75 Vyakhyans began on 4th of August 2021 and is being organized every Wednesday at 4 p.m. to celebrate the Platinum Jubilee of the foundation of our institution, that is PRL. The Vyakhyans are delivered by eminent speakers from India and abroad from diverse fields of science, engineering, management, etc. Today, we are honored to have a very distinguished speaker for the Amrut Vyakhyan, Professor Shiva Umapathy, Director, Indian Institute of Science, Education and Research at Bhopal. On behalf of Director PRL, my colleagues, and also on behalf of the Amrut Vyakhyan Committee of PRL, I extend a very warm welcome to Professor Umapathy. Many thanks, Professor Umapathy, for gracing our celebrations of PRL at 75. We are looking forward to your lecture on an interesting topic, that is Raman spectroscopy from physics to medicine. I would now like to invite my colleague Dr. Bala Shivraman to introduce the speaker to the audience and to request him to deliver the 74th Vyakhyaan. Dr. Bala, please.
Thank you, Professor Nandita. Good afternoon, all. It is an honor and pleasure to introduce Professor Shiva Umapathy, who is currently the Director of the Institute of Science, Education and Research, Bhopal. He is a laser spectroscopist and he was the chair of the Department of Inorganic and Physical Chemistry, shortly we call the IPC in IISc, and a professor of instrumentation and applied physics at the IISc Bangalore. He is known for his studies of molecular dynamics using Raman spectroscopy. Here, I would like to add, he is also the editor for the Spectrochemica Acta journal. There's a lot that we can keep on, telling a lot about Professor Umapathy because there's so much, but I will pick a few. Professor Umapathy graduated in chemistry from Kongunadu Arts and Science College of, which was under the University of Madras, and joined PSG College of Technology in Coimbatore, where he completed MSc in Applied Science in the year 1981. Then, subsequently, he moved to New Zealand his PhD and secured a PhD in physical chemistry from the University of Otago. And then several fellowships followed after that. Here, a few notable things. Professor Umapathy is holder of the Swarna Jayanti Fellowship in the year 2000, and then the J.C. Bose National Fellowship of the Year of the Department of Science and Technology in the year for the year 2010 to 2020. And he received the Young Investigator Award for the ICORS conference at the University of South Carolina in 1989, and Sir C.V. Raman Young Scientist Award of the Government of Karnataka in 2003. He was decorated with the Swanti Swarup Bhatnagar Prize, and many more things add to that. So, because Professor Umapathy is a very good speaker, we will save more time to hear from him. So, with this short introduction, thank you, Professor Umapathy, for accepting an invitation, and the stage is over to you.
Thank you. So, it's a pleasure to be part of this series of lectures. And I thank PRL and Dr. Bala for inviting me to give this talk. So I'll share my slides so that okay, so the title of the talk is laser spectroscopy from physics to medicine. It's basically using lasers to get information from various systems. And if you talk about light interaction with matter, there are many ways in which light responds to matter and the composition of matter. It can be absorbing the light, it can emit the light, and of course, it can also scatter light. Now, if you look at in this particular case, I'm only going to talk about scattering processes. You can also have reflections, but then let us concentrate on scattering. There are two types of scattering. One is your elastic scattering, and another one is inelastic scattering. The inelastic scattering is what Raman discovered in 1928, for which he was given Nobel Prize. So this is something I'm very proud that we are able to work what Raman started in 1928, close to 100 years later. His discovery now is going into commercial activity, including medicine and other aspects of detection of material in real life. So that's what I'm going to show you. In addition, I hope to show you that we can not only study about material and structure, but also we can see with respect to time how these change, and that is something that I'm going to demonstrate to you now. So let us say, for example, if you want to study about structures and changes in length and time scales, for example, a human species, you would look at them as 5 to 6 feet or 4 to 6 feet. And if you look at the next, that's what is the basis of time and space resonance spectroscopy. If the person is 2 feet or 3 feet, now the next level of imaging that is coming up to do a human imaging is terahertz-based imaging, which is still in the infancy. Hopefully, in the next 5 years, you will probably see technologies where you can image a human. But right now, using Raman spectroscopy, you can actually start looking at structures like these, which are tens of millimeter in size, which is basically a Drosophila. Drosophila is an outstanding model for medical purposes because the muscles are 70% genomes of these muscles are very similar to human muscles. Since muscle-related diseases are age-related, human age is 70 to 80, 90 years, and therefore research cannot go that long. So people use Drosophila as a model system, which has a lifetime of 60 to 70 days. So you can mutate different proteins and study them. So that's one of the studies that we have done to understand neuropathy and cardiomyopathy processes induced by muscles. The other science that we talk about more recently is studying bacteria, impact of bacteria on humans. This is about tens of hundreds of microns in size, and in 1 micron to maybe 5 micron, 10 micron. So this can also be studied in terms of spatial resolution using normal microscopy. And of course, when it comes to the real size that we are talking about, is now a few tens of nanometers, which is bacteriophage. And that's the next level of study that we must get involved in terms of trying to advance scientific research, in terms of fundamental research. So up to hundreds of nanometers, you can today commercially available technologies will allow you to go. Anything less than that, you need to build yourself. Of course, the ultimate imaging using light is probably looking at electrons, and which is about femtometer is the space limit that people talk about. So the next level of science in the world is going to be in understanding materials which are pico to femto sizes, which may have properties which are very different from our nanoscience and nanotechnology, which is the next level of science that people are now beginning to evolve as materials so that they can find unique properties using this. So, it basically tells you that one can actually look at materials in spatial domain and like to learn about the structures and changes in structures, chemical structures of these complex systems. Of course, some of them are time dependent. For example, what are the time dependent processes that you can see? The simplest thing that you all are familiar with is your splitting of eyes, where your eyes flicking is about 100 milliseconds per shot. So that is why your televisions work at 10 to 18 or 20 or 26 frames a second, because they're slightly faster than your eye response time. Eye blinking is about 100 milliseconds. Now, if in relation to that, if you look at our real life processes, look at protein folding, for example, is hundreds of milliseconds. And so they can even monitor protein folding if you can actually do experiments in the millisecond timescales, watching the structure every millisecond. And that studies are being conducted. And of course, the next step is to looking at neuron signals, which actually send signals in microsecond timescales between the synaptic cell and the axons. And that's how they communicate. And if you go faster, what we can look at is molecular rotation and how molecules rotate from one form of the other. And hydrogen bonding is one example where you can look at them, which is about tens of picoseconds. And of course, the energy relaxation of vibrations also happens in the time scale. But the most ultimate in a vibrational structure, chemical structure, is that if you look at these molecular vibrations, each of the vibrations are in femtosecond timescales. I will show you a bit more about this in a minute. So the idea is that if I can carry out experiments in femtosecond timescales, I should be able to watch these bonds vibrate in real time. So every time it vibrates, you will get a signal of the scattered signal. And therefore, for each vibration can be measured. And that one example I'm going to show you of what we do in our institute in Bangalore. So, as I said, we are now looking at vibrations. I'm sure all of you are very familiar with the infrared absorption spectroscopy. And Raman spectroscopy is very similar to infrared, except in the infrared is an absorption process. You put a light, it's like a UV visible electronic spectrum, where you put the light get absorbed. The difference in the light is measured as the frequency absorption. But in the case of Raman, what you do is you actually send a light and measure the scattered light that comes out. That contains the required signal of the vibration frequency. And the advantage is that the signal, you can increase the signal by increasing the laser intensity. And of course, you can monitor extremely low frequency at high resolutions using Raman, which is not so easy in the infrared. The other problem is that, for example, molecular vibrations are different types. Each one of them have their own typical vibration frequencies. Although there are 3 atom systems here, as you can see in water, you will see that there is, although there are 3 atoms, stretching itself, there are 2 types of stretching. One is the symmetrical stretching, another one is asymmetrical stretching. Both of them have different frequencies, and we must measure them at reasonably high accuracy. And that's what Raman spectroscopy allows you to do. So the other important thing that happens whenever there's a time-dependent process that happens in life, there are changes in energy of the bonds that you're looking at. There are chemical bond changes, breakages of bonds, forming of new bonds, and of course, a stronger bond becomes weaker. So I'm just giving you an example for students who might be listening to this lecture. For example, CC single bond or CC double bond you talk about in graphenes and carbon nanotubes, you will see that the CC bonds are weaker, therefore the corresponding frequency is also low. And CC double bond is stronger than CC single bond, therefore there's a corresponding increases. By looking at these frequency increase in wave number units, you can say that bond is strengthening or weakening. So with respect to time, if you're measuring something, you can actually say whether the bond is breaking or the bond is getting stronger. And similarly, for example, if there are new bonds are formed with different atomic weights, you can see here CH stretching frequency is about 3000 wave numbers. But if you change the H to D, then the frequency comes down to 2200. If you go down to C, then it comes to 1200. So the lightest atom has the highest frequency. The heavy atoms have the low frequency. So this is the other way to understand processes. So we have been working on this for the last 35 years. So we kind of have a reasonably good idea when I see a frequency, what it could be doing with respect to the system that I'm studying. So I'm going to talk about now femtosecond spectroscopy and what we learn in this. This is not something commercially available. We need to build equipment to do these kind of experiments. So we buy lasers, detectors, spectrometers, and of course amplifiers, plus oscilloscopes, frequency generators, and then set up a lab. And that's what we have done. Why is this interesting? Now, I can give you 2 examples that you can relate to. One is, for example, bacterial adoption or retinal. You know your eye responds in extremely fast timescales, but we have no idea how it works, right? Now, so when I look at a color, a green or a blue, my eye immediately sends a signal to retinal. Retinal then sends a signal to the brain saying what you're looking at is blue color, this part is red color. Now, how fast the retinal is moving is something very neat to understand. And also, why does it do it that way? Is there any way we can understand how these colors and the way it responds are controlled? And what is the energy involved in terms of your eye response time, in terms of your retina response time? The phase, I would leave it right now. This is for extremely fast processes where we study how a phase space of a scattered light is actually shifted because of processes in the system. And of course, we need to understand how interactions within and externally in a molecular system happens. For example, if you take graphene, is it a single layer or multi-layer? If it is a multi-layer graphene, what are the effects? And if you take a protein like hemoglobin, you can ask the question whether it is the protein matrix has any issue with the dynamics of the rate at which the hemoglobin absorbs oxygen and releases carbon monoxide. So those kind of questions need to be asked, and these are all happens in tens of hundreds of femtosecond or a picosecond, and this can be observed. And finally, the role of environment. The role of environment could be anything, even starting from a simple process that I'm going to show you today, is about how fast a molecule can move when I actually have an extremely viscous liquid. And therefore, can we control the process of chemistry or physics during a process? That's the other interesting question. So this is an area which is upcoming, which is all over the world. People are trying to understand various processes in these femtosecond timescales, and that's not very easy. Now, if you look at, for example, the carbon-carbon single bond, if we say 1300 as a frequency, and if you convert the frequency into time, which you can straightforward do, which is about 10 femtosecond. That means in 10 femtosecond, you have carbon-carbon as vibrated only once. Now, if you do experiments every 10 femtosecond, you can actually watch the carbon-carbon bond vibrate every 10 femtosecond. Therefore, you'll see a signal every 10 femtosecond for that frequency. By understanding that, you can learn a lot about various processes like energy flow, phase change, bond making and breaking. And of course, we can understand function of materials, whether it is conductivity in plastics or a lattice structures, or if you're looking at, for example, biological processes like bacterial adoption, retinol, or even hemoglobin cytochromes. So I'm going to talk briefly in the next 10-15 minutes about femtosecond Raman spectroscopy, how it is done, and of course, what kind of signals can you get. If you're a physicist teaching classes, where we talk about Heisenberg's uncertainty principle, and we always say that if you fix time, then you can't fix frequency. If you fix frequency, you can't fix time, which is actually the limit of what we learn from Heisenberg's uncertainty principle. That means if we can actually fix a time of 10 femtosecond, time 10 femtosecond is the theoretical limit, transform limit, which is time frequency is in 1300. That means, as per the Heisenberg's uncertainty principle, you cannot reduce the frequency width that is shown here to below 1300 wave number. That's a theoretical limit. So how do we then study? Because Raman spectroscopy requires experiments at 5 to 20 wave number resolution, because there are lots of frequencies which need to be understood, which are separated by 5 wave numbers. But then if the bandwidth itself is 1300, your scattering theory tells you the scattered frequency width is equivalent to the input frequency width. If my input is 10 femtosecond, which is 1300 wave numbers, the output also will have frequency width as 1300 wave numbers, which means any frequency which are separated by 5 and 20 cannot be understood, cannot be seen in the experiment. So I will demonstrate this here. For example, if you take a nanosecond pulse, the theoretical limit is 0.013 wave numbers at 790 nanometer. Now, if I record an experiment, if it is only 5 wave number width we are separating, you can see the bandwidth is about 0.013. Therefore, I can separate these frequencies very well. But if you go to picosecond, what picosecond is about 13 wave numbers. So therefore, this bandwidth that of the laser pulse is 13 wave number. The scattered frequency also contains minimum 13 wave number. And they can see that the bandwidth now has become much bigger. But of course, if you go to femtosecond pulse, no question here is so broad that you lose all the information that you have here in this system. So how do you do the femtosecond experiment? This has been one of the issues. So we started almost 15 years ago. As early as 95, I had proposed doing these experiments. But of course, we managed to get funding in 2005. And so we built up an experimental setup. The principle is very simple. If you're looking at linear scattering, what you do is you actually get this kind of light goes up and then gets scattered. And then, of course, the Raman scattered light is measured. But what I thought is, if we can do a stimulator Raman experiment, whereby if you can do at the same time, the same speed at which the scattering occurs, I can bring another pulse, which is femtosecond. And so this is a femtosecond pulse. Let's say this is a picosecond pulse, which has a frequency width much smaller than the frequency width of this. Then I can force the frequency that corresponds to this, the energy difference between the two, and which is the energy difference here. Therefore, you force the photon to come down. And so this is called the stimulator Raman scattering. So we decided that why don't we use this? Very interestingly, in the 2003, 2005, one or two people started publishing, and we are the third people in the world to demonstrate that it is possible to measure femtosecond Raman scattering as early as 2005. The principle is very simple. It's a third order nonlinear process. If you're familiar with, you will figure out that if you say the wave vectors, then you will actually get the signal in a particular angle. By measuring, you will be able to say what the signal is. And so what we do typically in an experiment, we actually use a femtosecond pulse. Of course, when the scattered light looks like this. But if you do a femtosecond pulse and a picosecond pulse, which the width is very small, with 30 wave numbers, so we then look at the frequency that correlates to the material frequency. Let's say, for example, if this is the frequency of the material, then these two photons interact. Therefore, what you see, a stimulated Raman scattering at that point. So like this, we can actually measure multiple coordinates. So for example, if you have lambda 1 and lambda 2, you can actually measure them at the same time. What you will see is two frequencies. Now, if you measure the signal on the anti-stroke side, what you see is inverse Raman. And if you actually measure them on the stroke side with respect to your excitation, you will actually see the gain spectrum, which is the linear spectrum. So this is, for example, now what I can do, now that I know how to get a signal, what I do is one experiment with just the white light, another experiment with the picosecond pulse. You subtract the two, what you get is finally a spectrum. And that's what we measure. So as I said, finally, we made a presentation of this into the ICOS 2008 in London. And on the same day, there is another group from UCLA presented the same data, which was exciting because, we are in line with the rest of the world. And of course, we are number 3rd in the world to demonstrate that you can do femtosecond Raman spectroscopy and also understand molecular structures using scattering of light. So now I'm going to show you how we set up in the lab. This is all set up by our students in IISc. This is like playing like a Lego. All bits and pieces are available. You can put them together. As I said, we need 3 laser pulses. So you cannot bring 3 laser pulses from 3 different laser systems because the light travels so fast, the frequency matching will be difficult. So you use 1 laser and then convert them into 3 separate sections. You can see here, for example, there's a beam that comes up. This beam is now goes into what is called a delay line that controls your time of arrival of the laser pulse, and then it goes to the sample. Now, similarly, 1/2 of the pulse is then transferred and then given here to measure to see what the pulse length is. This is a calibration unit. We generate a broadband white light. So here we have by using a Fourier transformation technique in optics, we are using 2 grating and a slit. We can generate the picosecond pulse. Here we have generated a white light pulse. So by combining these 2, we can stimulate Raman. If you want to study excited states, that is femtosecond processes, you have to create the excited state to initiate the photoexcited process. So what we do is we create 1 more pulse. You can see which is separated now coming out here. We split the pulse into another half. This pulse comes up and then converted into a second harmonic crystal. What you see is a 3rd pulse. So these 3 pulses are brought at 0 time in the femtosecond timescales. So you can see here the sample is flowing from the top to the bottom. And you can see, first we use the photo excitation pulse, which excites these molecules to the excited state. Within tens of femtoseconds, we bring the white light and also this picosecond pulse together and matches on the sample. What you get is finally is the signal that you measure. So I'm going to show you one example of how we're trying to understand molecular structures respond to light. So what you're seeing here is standstill beam. This is actually similar to the molecule used in your compact disc. Now, this is if the 2 benzene rings are on the opposite side of this double bond, this is called the trans-Philbene. If both the benzene are on the same side, for example, this benzene group comes back to this side, that will be called the cis. So the cis and trans are called the binary in computers. So you can write, say, switching between 0 and 1, you can actually write information. So this is a digital information storage methodology. So the speed at which this moves from cis to trans is a big question, because the rate at which you read information or you write information is critically controlled by processes like this. So the question was, people are asking at that time, how fast can I switch? What are the factors that control going from a trans to cis? So we tried to study them. So I'm going to go a bit fast because of time running out. I'll go to the results. So what we do is we create, for example, an excited state which has a lifetime of 85 picoseconds. And of course, there's a decay of the vibrational energy, which is about 11 picoseconds. This has been done by many people. We have to prove that we are actually in the right direction. So we did the experiment and demonstrated that this data is consistent with what other people have done. Then we do a femtosecond experiment. You can see here now, we start with minus 50 femtosecond, where there is no signal. And then we start going into 1.1 picosecond every 150 femtosecond experiment. And so we then look at the bandwidth, the rate at which these moves, and then do some analysis. We also do some computation to figure out what these frequencies mean. And so we know what components of the bonds that we are actually looking at. And based on that, we actually arrive at a data where we compare the way in which these frequencies move. The idea is that if you look at the peak position of the CC double bond, it has an oscillation. So this is something was unusual. We repeated this experiment many times, and we found this oscillation may correlate to something. So we converted into frequency, and that frequency is about 220 wave numbers. If you actually do a Fourier transformation of this power spectrum into time domain, the frequency domain to the time domain to frequency domain, the frequency you get is 220 wave numbers. So that means we are seeing a bandwidth oscillation of 220 wave numbers. So then we look at what is this 220 means. It is already known that 220 corresponds to an in-plane torsional motion. So which means that there is a motion of moving of these two double bonds across the double bond, the two benzene rings trying to torsionally move from one side to the other, from trying to go from trans to cis. That means the double bond has to kick the movement from one side to the other. And that is 220 wave numbers. So that 220 is affecting the CC double bond, and that's what we have seen in real time. So now we understand that the solvent acceleration of frequency pushes these 20 wave numbers, and therefore accelerates the whole dynamics of formation of these cis into trans. So of course, these are the various advantages of doing femtosecond spectroscopy. I'll skip that right now. So now I'm going to show something about biology, something on fundamental research, and then to applied research, and finally a new technology that we have developed, which has potential applications in many areas. So we do, these projects are carried out with my colleagues, both in Indian Institute of Science and hospitals nearby, Newhan's Hospital, which is a mental brain research hospital. We also work with Cancer Hospital, and then we have Ramaya Hospital, which is nearby. So it's a big team that works on these projects. So we look at, for example, tissues to look at cancer tissues to see is the cancer is going to recur or not. Can you identify the grade of the cancer from grade 4 to grade 3 or grade 2? Such studies are being done. And we also look at drug cell interactions. I'll show you one example of how a brain cancer drug can be studied to see whether its efficacy is good or not. And we also study liver injury to look at paracetamol. There's a tendency for people to take a lot of paracetamol. So there is a question about what is the level of injury, how fast can you study? We have studied that. These are all already published. You can look at my publications list. You will get some of these papers. Sepsis, I'll talk about today. Nerve injury is what I was talking about, using neurons, where you can see that how neurons can be monitored as soon as the nerve injury occurs, how the neurons respond to it. Which we have studied using cockroaches. We have looked at muscles using Drosophila, which I mentioned to you at the start of the talk. And we also look at bacteria. Here we have actually looked at many things, including identifying TB bacteria, then identifying harmful bacteria versus non-harmful bacteria. Right now, we are actually working on antimicrobial resistance, AMR, of bacteria, antibiotic resistance of bacterial diseases is a major problem today, and we are working on it. And I'll show you one example of some new technology that we have developed. What is the principle behind all of these? One has to understand light penetrates to different depths depending on different wavelength of light we use. So depending on what we want to study, whether it is blood, whether it is skin, whether it is arteries, or any part of the body, one has to understand these basic principles before we apply them. You can also ask the question that, Raman spectroscopy gives you so many frequencies. When there are too many bonds in protein structures or biology, how do you identify? Luckily, a lot of people have been working on this since 1970s, trying to understand biological proteins and structures. So there's large amount of information available to you. For example, here what you're seeing is alpha helix and also beta sheet structures. And if you look at the spectra of alpha helix and beta sheet, they're very different when you record them in the Raman spectrum. So by looking at their frequencies and activities, we can tell you what is the ratio of alpha helix to beta sheet in a particular protein. And if the ratio is changing, then you can talk about denaturation of proteins and therefore their application. Give you one example of, for example, a cell. If you take any kind of cell, a skin cell or a cancer cell, what you will have, you normally have a protein, you have RNA, DNA, and lipids, and of course, some other components which I'm not showing here. By recording the cell spectra, this is what you get. And of course, biologists can lyase these cells into different components as protein separately, RNA separately, and DNA. And then we can record these together. And then all of these can be combined to see the spectrum. So you can understand by looking at these changes in different frequencies or by lyasing the sample, by taking different components, you can actually understand the changes in chemical composition of any biological material. Why is this important? In diseases, what happens is normally first the chemical changes starts occurring in humans, and the chemical changes, when they exceed a certain threshold, then it becomes a symptom of a disease. So if you can start monitoring the chemicals in your body of different types, whether it is blood, plasma, serum, saliva, or urine or sweat or tears, any body fluid contain large amount of information of your body quality. And that's the principle we are trying to use here, trying to understand what happens. Now I'm going to show you one example of drug efficacy. Here I worked with Professor Somasundaram, who works with people in mental hospital where they give you samples of brain cancer cells. The question is that they use something called the drug, that the chemotherapeutic drug is called HDAC-I. H means that histone, D is deacetylase, DA is deacetylate inhibitors. So DAC is deacetylation inhibitor. What does that mean? You can see here. The histones are actually the core proteins, and the DNA actually coil around them. The role of there are different kinds of histones. And the brain cancer people have already studied using biochemistry and medicinal biochemistry that this is an important activity. So the histone structure, the part of the structure is shown here. So this is called acetylation process, the CH2 and CO. Now, or CH3CONH. This is acetylation. Now, the acetylation and deacetylation process must be maintained at all costs so that there is no damage to the cell. But when the acetylation then goes up, when the deacetylation starts happening too much, then what happens? Your cells become cancer cells, and therefore it's difficult to control. So they need to put a drug which can stop the deacetylation process. That is why the drugs are called histone deacetylase inhibitors. It inhibits the deacetylation process, so you reduce the reduce the formation of cancer cells. So this is what doctors are doing today with various drugs with respect to brain cancer. Now the question was, can we actually understand this process very well so that you can help doctors to decide which drug is good for which kind of cells? because some people are responsive to some drugs. Some people are responding to different drugs. There are different kinds of drugs that are used. For example, here, you have valproic acid is one drug, sorbitol is another one, temozolomide is another one, apixaban is another one, certinol is another one. So there are, you can see already we have 5. Now there are much more. So doctors can decide which drugs are good for you, but they cannot decide a priori unless they can see an experiment. So what we have done here, we try to actually incubate these cancer cells in different drugs and monitor the quality of the formation of acylation and deacylation process. If the deacylation is stopped, then we say that this particular drug is good for this cell. So we have done this in many different cell lines. I have done 5 cell lines here. Now each of the cell line corresponds to different metabolic person. So U343 can be one patient, U87 can be another patient. So you can try all the drugs. So now we have developed a drug screening methodology. In 48 hours, we can tell you which drug is good for a particular cancer. So you take a biopsy, separate the cells, and then put the cells in different units and try different drugs and see which drug is most efficient for that particular cancer cell line. And that's what they did. Now, what is more important for me to tell you here is that although this is a regular routine for them, I told my colleague that in addition to deacylation, we are actually seeing one more CH2, which is called propionization. And first, my colleague told me that in the biochemistry of understanding of glial cells, brain cancer and HDAC inhibitors, there is no history of propionization. I said, I'm measuring it, more than once, so there must be something. So he said, okay, let's go and try an experiment of a propionization based assay to see is there a propionization happens. And to his shock, he found out there is a very strong propionization present even in their biological experiment, which many people didn't notice it. So now actually the drugs need to be upgraded to also to depropionization in addition to deacylation. This is an example of how spectroscopy can help biology. And of course, we can develop technology to monitor health of people. So this is what this experiment has demonstrated. These are published. You can look at them in the literature. It is available in details. So we can monitor using this technique, a reliable, cheaper way instead of doing antibody assays, we can do a much easier drug screening and cell line screening. Now I'm going to show you one more example where we ask the question, can the light penetrate? How far can light penetrate into a system? Now, for example, one of the questions today people are asking is that, can we see what is inside bottles? You know that in the airports, they're stopping you from carrying water into the airports. One of the reasons is that it's possible that you may be carrying explosives or drugs. So then also you can ask the question is if you have tumor inside your body, can you actually see what is inside? So how long can you penetrate into the system? So what do you need to understand? So we go to the fundamentals of light. The question is how much light can diffuse into the body and what is known about light diffusion into body or into tissues. And of course, atmospheric scientists working on light will tell you that you can actually penetrate light through sky into the clouds through turbid, which is in the possibility of dealing them as a turbid media. And therefore, if that is the case, we should be able to do it also. It's also known the near infrared, like 1,100 nanometer light is used to monitor the oxygen levels in brain by absorption technique in babies. So which means we are talking about at least 10 centimeter, people are actually monitoring light absorption. If absorption can happen, if a propagation of light through turbid media through the sky and cloud goes, why can't I see scattering? Why can't I see Raman scattering at these distances? And that was my interest. So we started looking at how we can do that. So principally, we started doing some modeling and simulation. The idea is very simple. You put a light here. You have, that normally 1 in a million photon is Raman. The rest of the photons are Rayleigh scattering. The Rayleigh scattering is equivalent to the incident beam. Therefore, that light hits the next particle. So you can actually accelerate continuously by heating light here. And therefore, the green ones are the Rayleigh scattering. The red ones are the Raman scattering. So each particle can scatter light, and the green light can actually again scatter and hit the other particle. Therefore, you should be, in principle, you can measure. So we have done Monte Carlo simulations, and you can see, it's all published already. So we have shown that it should be possible to measure light in all directions if there is enough photon that goes into the system. And so we did that. So how do we now prove in experiments? So we did a 3-layer system, a cuvette, 2 cuvettes attached, and then 3 cuvettes attached, and then this is then made into a vertical cuvette. So we did an experiment like this. So you have 2 cuvettes attached. I have 2 different chemicals. One is dichlorobenzene on the top and transtylbene at the bottom. So if I actually put a light on transtylbene, you'll see the transtylbene signature is much larger. If I measure the light here, the light diffuses from the bottom to the top. And therefore, I can also see the signal of dichlorobenzene. If I actually go somewhere in the middle, you see the intensity of both of them are exactly same. Dichlorobenzene signal from the top, transtylbene from the bottom, approximately similar. But if you put the light on dichlorobenzene, then you can see the signal here. So these are all now well demonstrated, patented. And so what we did was we demonstrated we can actually start seeing what are inside these bottles. And therefore, without opening these bottles, we can tell you what is the chemical hiding inside. Then we went on to see how far can the light travel. So this is about 3 to 4 centimeter long. So we said we can go up to 4 centimeter. The beam is only 100 micron. 100 micron laser light penetrates into 2 to 3 centimeter on the top, 2 to 3 centimeter at the bottom. So we can measure Raman scattering in all directions. So this is what I'm showing you. We have patented all these. And this experiment is the crucial one, where we have taken a chicken breast, which is about 10, 12 centimeter long and 5 centimeter diameter. And we actually drill a hole and hide a chemical inside. And then we put laser light and push it through. Light is exposed on both directions. The light comes out on all directions. And that is measured and tells you. You can then reconstruct and tell you what is hidden inside. And you can see here, the chemical that we can see is exact size and location can be identified. And this is what we have done right now. So it can be applied in security, for example, finding explosives, narcotics. You can actually look at in materials, whether it is diamonds, petrol, silk, all kind of purity of materials. And in forensic science, in many applications of all kinds of fingerprints and various other issues. And pharmaceuticals, raw materials to quality control and process control. In biomedicine is predicting diseases is a big part, which I'm going to show you one example here. We have already demonstrated this technology. We have now created 2 prototypes. One is to identify bacteria, which is on your right, which has been given to the DRDO lab, DBEL in Bangalore. You can see they put the bacteria here and push it inside, and it's a touch screen. By putting a touch screen, you can actually see what is the bacteria inside, and what is on the top, it will tell you what is the chemical you are looking at. Here, for example, we actually have a system which is given for explosive detection, which is the same basic principles are very similar, but it can go up to 1 meter, minimum to 2 meters. At a standoff distance, you can actually measure what is hidden inside the bottle. And this, for example, is released in March 21 by DRDO Secretary and Chairman to look at what is hidden inside a box. And so that's something we have demonstrated already. So the final application, which is now becoming commercial, in the next 5 minutes, I'm going to show you. Now, what happens is because we have doctors working with us, one of the doctors told us that, he's a surgeon, when he said, when we do operation to a patient, after the operation is over, we take the patient back to the ICU and monitor them for a few days. Now, what happens is after a day or two, suddenly the patient develops complications. And we don't know what the complication is until the symptom starts appearing. One of the symptoms is infection-like symptoms, and which is basically changes in blood pressure, heartbeats and changes and all that. But that can also happen for many other reasons. But then they start seeing that organ starts failing, kidney is not functioning or liver is not functioning. Then they suddenly suspect, oh my God, it might be sepsis, it might be infection, which then they send a sample of blood samples to the culture test. And that test they can take 24 to 48 hours depending on the cultural media or culture system. And therefore, 48 hours later, they get to know that it is infection. Then they have to start treating them with the right kind of um, antibiotic. The problem is that, although when they see organ failure, they start treating them, assuming that it is going to be sepsis. But that's already too late in many cases. And sometimes when they get confirmed, then the dose of antibiotic also is increased depending on the severity of sepsis. And if you are not very careful in identifying it, sometimes it leads to septic shock. This is for patients coming from outside. People, they stay at home with infection and suddenly organs failure, and then they go to the hospital. The doctor then start measuring. Since they already have some antibiotic, they're not able to identify whether the person is infected or not. And then within another day, they actually go into a shock. When patients go into septic shock, almost 80% of people die. Very, very rarely people survive. And therefore, there is a need to identify these diseases much before these symptoms appear, much before the organs start failing. And can we do something? So we started this work in 2012 with Professor Dipankar Nandi using animal models with one of my students. And we actually started looking at different parts of the body, different parts of body fluids, different parts of dissections, from the spleen to blood serum. And we measure the signal of red blood cell, white blood cell, blood serum, all these, and learned that by monitoring blood serum, we can actually predict sepsis 24 hours before the symptoms appear, which means that as soon as they can inform the doctor that the person is infected, they can start the treatment before the organ starts failing. So therefore, they can save the patient very easily. So this was the idea. So we did this over 4 or 5 years. Then we actually took human samples. So I'll show you here what I just told you. The blood culture has been used for more than 50 years. And the only way to detect is organ failure test. And they also monitor lactate levels and the CRPs to see whether the person is heavily infected or not. In our case, what we do, we take a blood plasma and we measure them. Taking a blood, after that, within 2 minutes, we spin it, we take the plasma and we run the experiments for 1 minute. Within 10 minutes, you will know whether the person has sepsis or septic shock or healthy. And that's our demonstrated experiments. So the way we do it is we put the laser light, measure large number of spectra, and the spectra is all then curated, which is then used normally multivariate analysis, and then separate them into whether it is correct or not. But what we have done in the last 5 years, since 2018 or 2016, is that we started using artificial intelligence approach to collecting these data. The idea is that you get large number of data sets. You teach the computer about a disease versus non-disease. And we use a deep layer method called ResNet, which actually then identifies various kinds of unique variables for different diseases or different products. Then the computer, then if you give an unknown spectrum, and then based on what it is learned, it can come and tell you, it can predict what you're seeing is a particular material or not. And so this has potential applications in forensic, medicine, pharmaceutical, nanoscience and technology and defense. And that's what we use. So this is an example of sepsis where we have done this technology, where we have shown that septic shock and controls can be easily identified using this. So this has all been patented already. And so we have a prototype right now, which is actually ready to go into hospitals. We are working with 2 hospitals now in identifying these sepsis as a validation technique. Once the validation is done, then this will go into commercial stuff. I should thank all my students who did all the work in the last 20, 30 years. This is not the only set. There's almost, I think RP Singh can tell you that, there's almost 40, 50 people have spent a lot of time on various things. This is the latest group I'm right now in my lab who are actually doing all the work. This group, all of them have left. I don't think anybody is still around. I think some of you might recognize this guy is working for Horiba. Some of them you might recognize who are in India. He's in ISET, so many places. This guy you might recognize. He's actually in BRC, working right now. So this is a group right now in my lab who are running all the experiments. So I should thank all my students for their contribution. And of course, creative work doesn't come in unless they actually read and come back, make presentations, show results. So different discussions leads to more and more innovation and creativity. And I must support people who supported me in the biology side because I am not a biologist. So therefore, all these people, professors from microbiology, biochemistry, genetics, biophysics, genetics, and mental hospital. And Kidwa is a memorial cancer hospital, and Ramaya is another hospital. And I can receive funding from industry, Boeing, ITC, Unilever, Procter & Gamble as well. And I wanted to tell young students that, the frontier of science requires belief and courage. Because when you're walking into the unknown, you must be ready to face complexities and depressions. And when you know that that is part of progress, if you're positive and you have very strong knowledge in fundamentals, life is easy and you will deliver whatever you want. And let me thank you all for your kind attention. Thank you very much.
Thank you, Professor Umapathy.
Yeah, Bala, I can take a hello.
Yeah.
Yes.
Yeah.
Yes,
So we move to the next session. So before going to question and answer session, I would like to thank Professor Umapathy for giving a Vyakhyaan and covering the many topics, starting from laser spectroscopy, Raman spectroscopy, biophotonics, and some of the interesting applications like explosive detection, and even medical application. So thank you, sir, for giving huge information in a very short time. In question answer session, there are two kind of, we can say two part. First, we can invite our participants who are already at Webex, and then we can read a question from the YouTube channels. So I invite my audience, participants, viewers who are at Webex. They can raise a hand and ask question directly to the Umapathy. Yeah, Bala, please go ahead.
Yeah, okay. Just a short question. What are the limitations of Raman spectroscopy? Can you please tell us?
Limitations. Okay, so very interesting. So the limitation is that, since it's based on lasers, right? If you want to, for example, go into very long distances, the power structures have to be very long. Power has to be very large. So, that's that kind of where the intensity requirement is very large. Since we use lasers, it's probably not a good idea to use them. That's number one. Number two is the wavelength selection. If in a controlled environment like a laboratory, using a UV light is okay because you know what you're doing. But if you're going to go into commercial applications, UV is extremely dangerous, and it is not something I would normally people agree to do. So that's the other limitation. Although the most efficient way to do it might be UV laser, but then in practical purposes, it's not a good idea. So there'll be a limitation as well.
Okay. Thank you.
Any other question from Webex?
Harish Gadvi here.
Yeah,
hello.
please go ahead. Yeah.
Hello. Just when you are talking about the medical applications, I'm wondering, is there a Raman spectroscopy applications in diabetes, sugar detection? Because I believe they are still have to rely on to this pricking the finger and taking out the blood. For example, like oxygens in corona time, people can just measure it by putting the instruments on the finger, but
Right.
sugar. So, is there any development going on this direction?
Correct. Good. No, right. I'll tell you that right now we are trying. Many people are trying. One of the issues is the signal resolution at the near infrared is not very good because blood we can measure it in the blood using near infrared light. That is non-invasive. We can use fiber optics. So we have been able to measure as well. But quantification has been difficult because the signals are very weak. But I don't think we are far away because large number of new detectors are coming for near infrared. I think within the next 5 years, you will have something very similar to oxygen detector for sugar detection. I think many people in the world we have tried as well. My students have tried to use light penetration on your arteries and your nerves, and we go light and the light comes out and you measure the light and try to look at the sugar concentration. But quantification in terms of accuracy that you do with your existing technique, we are not able to reach there. That's because the detectors are not really more efficient. But I'm sure we will in the next 5 years. There might be some good detector come up with quantification, and we should be able to do that.
Thank you, sir.
Thank you. Yeah. Yes, RP.
Yeah, so just wanted to know, there are many techniques for this cancer detection. For instance, spectroscopy is there. Then you can do scattering, fractal dimension is there. Then you are saying Raman spectroscopy. So how doctors will decide which technique is good or which technique they should select? There are so many techniques coming for cancer this detection. So,
I'll
but
tell
I
you.
doubt
So,
still anything is going into the medical science.
So medical science are very mature technology in terms of adopting technologies. So there is something called receiver operating curve, right? It's called ROC. All the doctors know about it. The idea is, it should be 100% sensitive and it should be 100% selective. So if you plot sensitivity versus selectivity, it should be 1:1. And we're as close as possible to that. So any technique can be used to measure them, whether it is scanning, whether it is scattering, whether it is biopsy. So they actually look. So we have, even when we do that, we need to demonstrate that by measuring and showing our ROC curves and see how close we are. Right. So that is one way of making sure the doctors can choose which technology. Of course, different countries do. For example, personalized medicine is an expensive business, right? So, although it is more accurate and it can be done, it's expensive. So countries like India does not adapt so easily. But in Europe and US, they're starting to do. So that's it's also going to depend on the cost. That's what I mean.
Cost, yeah, mainly. Yeah, I think. Yeah, it will be great.
Yeah.
Thank you.
Thank you.
Any other question? Sir, may I ask a question? I'm just curious. Yeah, so like you discuss that some big molecules, biomolecules, using Raman, stimulated Raman or some techniques, we may get information. So my question is how to differentiate the degenerate frequency in Raman. Let's say two frequency are degenerate, same frequency.
Right.
So how to
that is
differentiate?
right. A very interesting question. So normally what happens is, luckily, right, molecules have more than one frequency. All frequencies cannot be degenerate.
Yeah.
One frequency could be degenerate, number one. The second thing is, when they have frequency, they also have amplitudes. These amplitudes are related to the symmetry of the system that you're looking at. So the symmetry also will change, which means that the intensity, amplitude of these frequencies will also determine the structure. Right. So you can actually either look at, for example, if you want to look at as a variable for statistics in terms of frequencies, you can look at not only change in frequency, but also you can look at relative change in the ratio of amplitudes of multiple frequencies. So if you have 12 frequencies, then you have 12 amplitudes. You can put permutation combinations and look at how uniquely to identify a particular chemical. So you can do
Yes.
that.
Thank you, sir. Yeah. So if there's no question from Webex, then I can read the question that posted from the YouTube. My colleagues Vineet has posted here. So Dr. Rakesh Kumar Misra, his question one is how Raman spectra used to analyze material properties, physical and chemical properties through it.
So
Yeah.
The Raman spectroscopy can only tell you what is the structure, how the bonds are connected, and if there is a change in structure because of some function or some process, pressure or temperature or something like that. No other physical property can be studied using Raman. Only Raman spectroscopy gives you only the structural information.
His second question is how UV destroy bacteria through use of light.
Right. Okay. So, if you know how to calculate energy per photon, right? The reason you and I can see light is because these visible lights have photon energies are extremely low. And so even if you put a single photon, we don't get burnt out because our body can absorb them and reasonably well. But UV light per photon energy is extremely large. So even single photon that hit your finger, you will actually like a needle touching your skin. So it's extremely high energy photon, and that is why they can kill anything. So that is why we don't normally encourage anything below, 250 nanometers or 280 nanometers is not safe.
Last question from YouTube is what are the importance ultra fast frequency used in biomedical engineering? I think he is mentioning that ultra fast laser used in biomedical engineering. Question mark, which types of instrument use?
Well, ultra fast and biology is still a long way to go from each other. So I'm not quite sure whether we are there anywhere close to application. A lot of people are trying to do imaging, for example, using ultra-fast lasers. There is something called CARS imaging. There is something called stimulated Raman imaging. They're looking at tissues. They're looking at membranes. Yes, they are good for fluorescence imaging. But of course, these materials that you're studying, the fact that you're talking about ultra-fast lasers means that the number of photons per square density is extremely large. So that can damage most of the sample. But then, things like membranes in CH, NH touching in tissues, they don't get damaged so easily. So those kind of vibrations can be easily studied. So the applications are yet to evolve. It is not there yet. But of course, you can use regular commercially available amplified or non-amplified femtosecond laser systems and put it through microscope. Today Zeiss and various Leica and various other companies are actually trying to sell imaging, fluorescence imaging using ultra-fast systems, which is basically 2-photon, multi-photon imaging processes.
Thank you, sir. There is no more questions. So now I request Navinda to take a lead.
Thanks, Rajesh. So so we move on to the next, the end of this session. So that is the vote of thanks. So it's my pleasant duty to conclude this session of the Amrut Vyakhyaan. It was a very illuminating talk by Professor Siva Umapathy on Raman spectroscopy. And it is nice to learn how the molecular dynamics can be visualized and explored in real time. Now I would like to say thanks to all involved in Amrut Vyakhyaan series. So first and foremost, on behalf of Amrut Vyakhyaan committee, I would like to thank today's speaker, Professor Siva Umapathy, for taking time from his busy schedule and for delivering this excellent Vyakhyaan and for patiently addressing all the questions and comments of the audience. Next, I would like to thank Professor Nandita Shrivastava for spearheading this series with her constant efforts and enthusiasm. Thanks to Professor Anil Bhardwaj for inspiration and guidance, and to Professor Palamraju for his guidance and efforts to keep the series going well. With this, we come to the very end of today's session. Now we will be signing off from this platform and hope to see you all next week. So probably in the next week, we generally have on Wednesday 4 p.m., but the next Vyakhyaan might be shifted to Thursday. In any case, we will let you know. So with this, we close, and I wish a very happy New Year 2023, and good evening to all of you.